It's a great pleasure to invite Alexandra Weyrich from Berlin and Leipzig. So right now you are working at the Integrative Biodiversity Research Institute in Leipzig. And so Alexandra did her PhD in 2007 on DNA methylation and histone modification in the spermatogenes of the fruit flies. Is that correct?
That's correct.
At the Chinese Academy of Science in China, in the Max Planck Institute that is based in China. And then she did many years of postdocs working on epigenetics effects and gene expression in wildlife populations. So she worked a lot at the Institute of Zoology and Wildlife Research in Berlin, where we met. So because I also did my postdoc over there, that was around 2015, 2016. I was there in 2016, so we met about that time. And then she was also, while being a postdoc, she also was a project investigator of many projects from 2018 to today, running projects such as epigenetics and stability and plasticity in wildlife population, and the interspecies epigenetic project at IDIV. And now you finally got, after all these years of postdoc, a permanent position at the Leibniz Institute in Berlin, the Wildlife Research and Zoology Research Institutes. So now today she's going to be talking about epigenetics, her main topics in the spotted hyena population. Please.
Thank you, Lucille. Thank you for the invitation. Thank you all for being here, of course. Today I will talk about epigenetic signatures of social status in free-ranging spotted hyena. So I guess none of you is working on spotted hyena, or few are working on wild mammals. I'm currently, as Lucille already said, I'm currently a postdoc still at the Synthesis Center in IDIV. It's a biodiversity center. I'm employed by the University of Leipzig. I'm working together with the Helmholtz Center, and all the research that I'm showing here was done at the Leibniz Institute for Zoo and Wildlife Research. What is the epigenetic signatures? We just published it. It's so it's just out, and and the first author is Colaviu, and also Sarah Benheim is the second author, who is the leader of the project, of the Spotted Hyena project that was funded by Herbert Hofer Marine East. So what is epigenetics? Some of you might know, I guess everybody heard about epigenetics already. So epigenetics is a field that studies the regulation of gene activity without a change in a DNA sequence. And epigenetic modifications are dynamic and reversible, which is in contrast to changes or to modifications within the genetic code, which are not really reversible. Epigenetic patterns that occur in the germline, like in oocytes and sperm, can also be transmitted to next generations. What kind of epigenetic mechanisms are there? So there is DNA methylation, histone modification, and small non-coding RNA for the moment that are regarded as epigenetic mechanisms. So DNA methylation is a CH3 group. It's a methyl group that binds in mammals, especially to CpG position. So to a cytosine, this is a cytosine. It binds here to the 5-carbon site and forms a methyl cytosine. And this is mostly when it's neighbored by a G, by a guanine, so a CpG position. If the CpG molecule is methylated in the promoter region of a gene, then the gene can be switched off. It's not always the case, but it can be switched off. And so it's basically like a switch to turn a gene on and off. So it can be, so it works in the regulation of gene expression directly on the DNA level. Histone modifications work on histones. So the DNA is wrapped around these histone molecules, we all know that. And the parts that are more open are more, can be more accessed by polymerases and transcription factors to start gene expression. Histone modifications can be methylation, but it can be monomethylation, dimethylation, trimethylation, so different types of methylation that are not methylating the DNA, but the single amino acids, like lysine methylation, for example. But it can also be acetylation, phosphorylation. So there are different other modifications happening here as well, and they all work on the compaction and the decompaction of the chromatin. Small nuclear, small non-coding RNAs are RNAs that work on, that they bind to the mRNA after transcription and activate dicer and argonaute, which cut down this mRNA into pieces, and therefore it's a regulation of gene expression on after the transcription, so post-transcriptional. And all these mechanisms are shown to have a function in the regulation of the gene activity. And this figure is actually from a comic that I once made. So I'll show you a bit later. So whereas epigenetic modification has been shown to be important, it's especially important in the early development. Like there are genes that are called imprinting genes. Do you ever heard about imprinting genes? No? Okay. So basically imprinting genes are genes that are activated in the offspring either by the father or by the mother. Yeah. So they are parent of origin activated. And this activation is controlled by methylation, by DNA methylation, by epigenetic marks. It's also very important in cell differentiation. Like you have eye cell, heart cell, different types of cells, but they all have the same DNA, but they all have a different function.
Someone turn off your microphone, please.
So they all have a different type of regulation. So different genes are activated, and they are also regulated by DNA methylation patterns. So different cell types have different methylation patterns. Also, there are transposable elements in the genome, like a lot of them.
What was the famous example of biological invasion?
Biological invasion.
Vous pouvez éteindre vos micros?
Like the yes.
There's someone online that has a microphone on and is speaking right now. May you please turn it off? So transposable elements can jump, can relocate in the genome. They can also increase their copy numbers, and this is not really, like, they are regulated also by DNA methylation marks. So they can be silenced by this. And it has been shown, and this is where I got really interested in epigenetics, is that they change the patterns of methylation of epigenetic markers, mechanism change in response to environmental factors. So there are several studies about this. And I have also been conducting several studies about this. So we always were looking into how an environmental change is changing the epigenetic pattern. Like, are there epigenetic modifications? For a long time, this was not really so well investigated. We did a couple of studies, for example, on guinea pigs, on wild guinea pigs, where we exposed male wild guinea pigs to an increase in temperature for only 2 months of 10 degrees. And then we investigated the same individuals before and after the temperature increase, as well as the next generation, as well as the son of these individuals. And we saw differences in DNA methylation patterns in the fathers and also in the next generation. So this is quite interesting also in terms of global changes. We also investigated nutrition changes, like, and also in the guinea pigs, also in a similar setup, and also found that also in the offspring there were differences between offspring that where the fathers were exposed to a change in nutrition. We also did a nice studies on the spalax, on different soils, and in cheetahs, in between captivity and zoo animals. And what I will talk about today is the spotted hyena, which live in a different, where in the same clan, they live in a different social environment. And here the question is, does the social environment, like the social status, translate into status-specific epigenetic profiles? And is this happening across age, like in cubs and in adults? Can we see a pattern, a significant pattern? Why did we use a model species, like why did we use the spotted hyena? First of all, the spotted hyena has been studied at the Leibniz Institute for Zoo and Wildlife Research for a very long time. They have been continuously monitoring the animals since 1987. So this is more than 30 years in the Serengeti National Park in Tanzania. So they know each animal by heart, by eye. They can identify them. These animals live in a special social organization, which is the linear dominance hierarchy. Here, every individual holds a specific rank. So you have alpha, beta, gamma animals. Therefore, you can group them into high-ranking ones and also low-ranking ones. And they live in large groups. The groups are a bit similar to many primates, and especially the female, and it's female dominated, and the females are philopatric. So the females stay in their rank throughout their life. Also, mothers transmit their rank to their offspring. So it's an inheritance of a rank in which high-ranking cubs receive more care from the mother and more social support, while low-ranking cubs receive less care and less social support. And they learn, the high-ranking ones learn to behave socially dominant and become high-ranking over the low-ranking ones, or over the ones that the mother behaves. The ones that the mother behaves dominant to, the cubs also can behave dominant to, and the ones the mother behaves subordinate to, the cubs also can behave subordinate to them. So this is basically a nice figure where it shows that like the tail up and the ears on the front is a dominant animal, while the ears on the back and the tail, like a bit like a dog, shows that it's a subordinate animal. So they always have these interactions. And why do they have this kind of setting? Well, it is thought that it's a reduction of costly aggression within a clan during encounters from the outside. So this kind of social status, this kind of dominance behavior is a very complex trait, or it's an emergent property, because it only exists in interactions, in dyadic interactions of animals, and it's not, the animal itself does not, like out of the clan, social status does not play a big role. So in this, this dominant behavior or the social ranks have a lot of influence, a lot of life history traits. Like, for example, they high-ranking ones have a better access to resources because they can forage in the close proximity of their den. So they have priority access to resources. Then they can also come back more often to feed their offspring, so they have a higher lactation frequency, and they don't need to commute so far distances. they have a better health, and it has been shown that they have an earlier onset of reproduction, so they have a better Darwinian fitness. While the subordinate ones have a lower access to resources, they have to commute very far distances, like up to 70 kilometers one way, to go hunting, and sometimes stay away quite a long time, like for several days, up to a week from the den. And therefore they cannot lactate their offspring so frequently, and the offspring is not growing as fast and also starts later in the reproduction. So what did we do here? We studied 3 different clans. They're called Isiaka, Mamba, and Pool in the Serengeti National Park in Tanzania. And they were all known, so they were observed for a long time. And when these animals are in their den area and they defecate, they go outside of the den. The researchers observing them can see which animal goes and go afterwards and pick up the feces samples. So these are the feces. And the feces samples in hyenas, they have a layer, they have a mucus layer, which is basically the gut epithelium cells which from the gut. And this is the material that we have been using in our DNA methylation analysis, or for our DNA methylation analysis. And it's the first time this kind of material has been used. So it's a non-invasive sample material. And this material is supposedly, it should be only hyena DNA. But as the gut is full of bacteria and food, like digested food, there will also be a lot of other DNA inside. And there's when we can make use. So we wanted to separate the DNA, the mammalian DNA from the bacteria and other DNA. And we also wanted to investigate only the ones that is methylated. So therefore we could use a method which is targeting only the CPGs that are methylated. And in mammals, it's mostly CPGs that are methylated. So using a method, which is a capture method, which uses an MBD protein. It's a methyl binding domain protein that specifically binds to this methylated CPGs, like to the methyl group in a CPG. And therefore we could, and this is then connected to a biotin. A biotin molecule really strongly binds with streptavidin, and the streptavidin was coated around a magnetic bead. So all this magnetic bead, MBD protein and methylated DNA were one molecule then, and could be with a magnet drawn to the side of the tube, and all of these were then methylated DNA. So everything that remains, you can just withdraw. You have all your non-methylated DNA and can only use on your methylated one, which is also enriched for the mammalian one. So this was really cool, working on feces samples, basically. So here then we have only the methylated one, and we go into next generation sequencing. Using preparing a library and then doing the sequencing. Here we use 150 paired end, base pair paired end sequencing. Then we use these reads that are all methylated DNA and map them to the genome. And therefore, when we started the project, there was not even a genome for spotted hyena. When we finished the project, there were 3, which was fortunate, so we could test them all. And also we had a collaboration with Shao et al. So we got their genome sequence earlier, and it turned out to be the best of them, which is, we were lucky. And yeah, and then we could basically map all these to the reference genome, and could compare the read counts of each, for each group. We could compare the read counts between the groups. And this is done by using a 300 base pair sliding window that slowly moves over the reference genome and counts the reads. And comparing high-ranking and low-ranking ones, well, for high-ranking, we had a number of 18 individuals, so it's not a big amount, but it's what we could do. And in this, we had 9 cubs and 9 adults, and we compared them to 24 low-ranking individuals with 15 cubs and 9 adults. And we found 179 regions that were specific, that were differentially methylated between high-ranking and low-ranking, and 26 of these were hypomethylated in low-ranking, so they had less methylation in low-ranking, and 153 were hypermethylated in low-ranking individuals. So there seems to be more methylation in low-ranking females. 42 of these DMRs of these differentially methylated regions of 300 base pair were overlapping with genes. And since we also did a custom-made annotation, we could also see, very often annotations just include genes. But we are also interested in promoter regions because these promoter regions, if they are differentially methylated, they can control the gene expression afterwards. So we made an annotation of the genome ourselves, and we included promoters, transcription start sites, start codons, exon, introns, so we could define it more specifically. And here we saw that intergenic regions have the most, so in between 2 genes, most of our differentially methylated regions are between these genes, and it's 137. And only 2 were in promoter, but several were in the gene regions. Then we also looked into, this is basically showing the same as I just said. So we have more hypermethylation in the low-ranking ones, and intragenic regions are less than we have a lot of intergenic regions and less in the intergenic regions. And if we find 2 differentially methylated regions beside each other, they become one bigger stretch, and therefore we can have from 300 to 600, like 2 together are 600, so the regions that are differentially methylated become larger and larger, which can have a biological meaning that we don't even really understand yet. Yeah, but as you can see here, we have up to 3,600 base pairs of differentially methylated regions, which is interesting to understand, but we did not yet fully understand it. Looking into the genes that we find, because the genes are the main regions that we understand yet. So we were looking into DMRs that are in overlapping with genes, and here we found we validated them by subsampling the 42 samples in 6 groups, and then we ran the analysis again. And if the methylation, like if suddenly it was hypermethylated, the DMR, and not hypomethylated anymore, we excluded this. So for all the samples, so that we don't have a sample effect in the whole analysis. So here we found that of the 179 ranked DMRs, 30 were not consistent and excluded. And the ones in genes, 42 ranked genes, these 42 ranked genes, ranked DMRs were overlapping 45 genes because they were partly very long. And only 1 was excluded here, which is this one, because it stretched over to be hypermethylated in one of the subsampling analysis. Then we also wanted to see what is the function, what is the biological function of these genes. And therefore we used a network analysis. We used all these genes that were differentially methylated. We included them in a network analysis. which is an open source string. And each of these dots here represent a protein. So the genes are encoding for proteins, so all of them. So the assumption is that if a gene is differently methylated, the protein will also be differently expressed. So there's even more, or either more or less of this specific protein. And each of these strings show that these proteins are working somehow in the same physiological process. Yeah. So here, for example, we can say we found, for example, this blue proteins to be differentially methylated in their genes. And they are all related to energy conversion, which is quite interesting. So there seems to be a difference in energy conversion between high and low ranking spotted hyena. They were also enriched in genes that are important for glutamate receptor signaling. These glutamate receptors, they build the axis between the gut and the brain and transfer information from gut to brain, which makes sense when we look into feces samples. We also found several genes that are important in the immune response, like these green ones here, which also would make sense because low-ranking ones have a lower health than high-ranking ones. And some of them were also in ion transportation, which could be related to the energy conversion. So these findings may reflect the access to resources, that what we have been, that was our inference on this, that they may reflect the access to resources because the low-ranking ones have to go up to 70 kilometers one way, as we said before. So they are long-distance runner. So maybe they have a very different, at least in the energy conversion, they have a different physiological system. This could be an explanation. They also give a first set of biomarkers. And of course, we would have to test them in a bigger population. And we could detect this all in a non-invasive sample material, which is quite nice. Then we also turned it around and we wanted to see, do our DNA methylation patterns predict rank? And therefore we used different variables in a random forest analysis. And we found that when we use rank DMRs, like the total number of rank DMRs, that we found we have quite a high predictive power to predict rank. The validated ones, however, are even better in predicting ranks. So, and the excluded ones were really bad in predicting rank, like 45% is not enough for a prediction. A lot of other studies before on hyena have been using mean methylation analysis. So this is basically just giving how much methylation is there overall in a genome and not in single positions. And as you can see here, at least for social status, it does not predict very well. Or not at all. Then we also looked into these mitochondria genes. Like as I told you, the gene, the conversion, the energy conversion genes, they were surprisingly all located in mitochondria. And so these are mitochondria genes because mitochondria are the organelle in the genome, in the cell that produce energy. So here we wanted to see if these genes are really in mitochondria or if these genes were also active, if these genes were expressing DNA, RNA. So if they were active in their reproduction. And for a long time, it was not even known that mitochondria were methylated. So we find mitochondria genes to be highly methylated, and they were even higher methylated than the ones in the nuclei. And then we also wanted to know, are these genes expressed or is there also a higher expression, or are they all deactivated? And we found also a very high expression of these mitochondria genes in comparison also to the nuclear genes, which were pretty lowly expressed. So this is an interesting, so it shows that they were also active and not numb. So in conclusion, what could we say? We can say that social status seems to go under the skin, and this happens across age. We can also say that we identified status-specific differentially methylated regions. They were mostly, or like many of them, were an energy conversion, which may reflect the access to resources of the high-ranking versus the low-ranking ones. They can give a first set of biomarkers, and they were even detectable in a non-invasive sample material, which we hope can also be used for other wildlife studies in the future. This project, of course, was not just this. Like we worked on epigenetics, which was the main mission of the project, but we also have other biome data here, like microbiome, eukaryotum, and we have also immunology data, and all this will be further analyzed and studied in the future. What else do I want to look at in the future? Like some future questions that I want to tackle or address are epigenetic modifications specific for specific environmental factors, or are they very different? Do molecular patterns of hosts can be used to predict disease susceptibility? Then also I would like to look more into the mitochondrial methylation, because the results that we have here are really intriguing. And I would like to study the role of mitochondrial methylation. I would also go more into, or what is always very nice to do, is to study the role of epigenetic mechanisms or epigenetic modifications in phenotypic alterations. And what I have been working on in the Biodiversity Center in Leipzig was tackling, or was the first start to see, can epigenetic modifications serve as an estimate to predict the adaptive potential in species? And the last one I have been doing for the last couple of months at least, because I have mostly been working on the hyena study, with Walter Durka from the Helmholtz Center of the UFZ, and he's also a member of IDEF. With Christina Richards, she's working on plants, mostly on epigenetics in plants, in the University of South Florida, but also in Tübingen. And also together with Mark Schmidt, who is a data scientist in Switzerland. And with that, I would like to thank you for your attention, and I'm happy to answer questions. And this is the comic that I did at some point, which I unfortunately forgot to bring you some copies. I would have liked to give you some. Okay, thank you.